Okay. The subject of my talk is impact of congenital heart defect on the developing brain. Sorry, but my screen is on my right, not in front of me. Uh, congenital heart defect, congenital heart defect affects approximately 1% of live births, making them one of the most common birth defects worldwide. Although survival rates have dramatically improved thanks to advances in heart surgical techniques and neonatal care, concerns remain about an increased risk of long-term neurodevelopmental outcomes. Initially, it was thought that children with complex congenital cardiopathy were at increased risk of neurodevelopmental difficulties, attributed first to brain damage resulting from heart surgery. Subsequently, an increasing number of studies have shown that prenatal imaging can detect brain abnormalities at birth, but even before birth. Nevertheless, mechanisms underlying the delayed brain growth remains largely unknown, as very few neuropathological studies have been conducted to evaluate neuropathological histological brain damage in subjects with congenital cardiopathy. This work was performed in the Department of Pathology of Sainte-Justine Hospital with three main objective. First, to identify brain lesion that occurs in fetuses with congenital cardiopathy. Second, to understand the mechanisms leading to abnormal brain development. And third, to correlate them with antenatal imaging finding. The study included a cohort of 40 fetuses, 21 male, 19 females, selected with the following criteria. Fetuses with cardiopathy diagnosed at ultrasonography and studied after termination of pregnancy due to a poor prognosis. Neonates, and second, neonates including preterm and term babies died in the first day of life, in most cases in the first day of life and without surgery. Syndromic cardiopathy with significant extracardiac malformations likely to affect brain development were excluded. The subjects were classified according to their gestational term into second trimester or T2 subject. They were 32, and third trimester subject or T3 subject. There were only 8. Despite the difficulty in finding true controls, we were able to select 5 second trimester control fetuses and 5 third trimester control fetuses or neonates. In each case, a complete autopsy was performed according to the protocol detailed by Langley 1971 and Wigglesworth 1984. Each brain underwent a detailed macroscopical and histological examination. The brain weight for each case was recorded before and after fixation and compared with reference value. Routine representative samples were taken in each case for histological examination, as shown on this diagram, according to our usual protocol of sampling for histological studies. Based on the finding from this sample, we completed our neuropathological study with Luxolfas blue myelin staining in third trimester subject and with immunohistochemical studies on selected samples, which will be detailed later. Congenital heart defects were categorized on the basis of ultrasonographic cardiogram performed in the second trimester by pediatric cardiologists and postmortem examination of the heart performed by pediatric pathologists and often in collaboration with pediatric cardiologists. Due to the particularities of fetal circulation, fetal cardiopathy could not be classified in the same way as congenital cardiopathy in living infants and children. Most cardiopathies in our series were severe cardiopathy due to the poor, due to an unfavorable prognosis, due to the unlikelihood of corrective surgery, leaving only palliative measures. Cardiopathy heart defects were as follows. In second trimester subject, hypoplastic left heart complex was the most frequent. We have 15 cases out of 32 because always of the poor prognosis, as parents in most cases opted for termination of pregnancy. Other cardiopathy were tetralogy of Fallot, hypoplastic right heart complex, double outlet left ventricle, univentricular heart, Epstein anomaly, transposition of great arteries, pulmonary valve atresia with intact septum, and atrioventricular septal defect, with 1, 2, 3 cases in each category. In third trimester subject, we observed also 2 out of 8 cases of hypoplastic left heart complex, and the other were aortic heart coarctation, Epstein anomaly, transposition of great arteries, double ventricular atrial discordance, and dilated myocardiopathy, with 1 case in each category. Macroscopical and histological examination was performed in all cases except 3 cases because placenta were not sent to the Department of Pathology. An array CGH study was performed in 34 cases, but not performed in 6 cases, including 2 fetus and 4 neonates, due to parental decision. For statistical analysis, subjects were classified according to their trimester of gestation, second trimester, third trimester. Due to the difficulty of precisely of quantifying microscopic abnormality on a postmortem material, we used a binary system to assess the presence or the absence of lesions. To determine if a given category, CHD category, was associated with a lesion, we performed Fisher exact test for each region of interest, and to determine if a given period was associated with the presence of lesions, we used the same test, and we used also the test for fetal sex. Placental weight, when studied, was in all cases in normal range and without histological lesion. All placenta were normal. Array CGH study detected genetic anomalies in 3 out of 34 cases. These anomalies were for the following anomalies. In a 22 fetus with double outlet left ventricle, we found deletion 15q11.2. In 23 fetus with tetralogy of Fallot, we found a deletion 5q35.3, including FLT4 gene. And the mutation of FLT4 gene have been previously reported in subject with tetralogy of Fallot. And in a third trimester fetus with aortic arch coarctation, we detected a duplication 3p13, which include this gene, RYPB gene, and the RYPB gene is known to have a significant role in the development of cardiac progenitor. Our first objective in this study was to identify brain lesion that occurs in fetuses with congenital cardiopathy. At the macroscopical examination, the brain weight among all the second trimester subject, was at the 50th percentile and was in agreement with the fetal weight in all cases except one case at the 5th percentile without histological lesions. In 5 out of 8 third trimester subjects, the brain weight was at or below the 5th percentile, but in these fetuses, the body weight was in normal range, not correlated to brain weight. Our neuropathological study showed that 4 structures were often affected in utero in subjects with congenital cardiopathy, namely hemispheric white matter, subplate, germinal zone, basal ganglia, but no significant lesions were observed in other brain structure, the hippocampus, the thalamus, brainstem, and cerebellum, and no cortical malformation was observed in any subject. Changes in white matter were observed in only two second trimester subjects, including a preterm baby, a 26 preterm baby, and in 7 out of 8 third trimester subjects. White matter lesions were diffuse, and they consisted in a microvacuolation, as you can see here, associated with diffuse astrocytic gliosis and activated microglia, well demonstrated with immunohistochemistry. In the injured white matter, we observed a density of oligodendrocytes identical to that of control, but numerous oligodendrocytes showed pycnotic or apoptotic nuclei. In the injured white matter, in all subjects with injured white matter, we observed also axonal lesions, and these axonal lesions were both quantitative and qualitative. Here you can see a fetus with congenital cardiopathy. Then you can see that axons, the density of axons was severely reduced compared with control. And the axonal lesion consisted in fragmentation of axon and swelling, as you can see here, of axons. Axonal lesions were observed mainly in the crossroad areas and areas in the white matter where motor, sensory, projection, and commissural pathway cross. Changes in white matter led us to study interneuron that migrate late from the germinal zone to the cortex in the third trimester. We focus on interneuron expressing calretinin because they are the most prevalent in the prenatal period. Compared with control, here a third trimester subject with cardiopathy, and here an age-matched control. Compared with control, calretinin positive interneuron were reduced in the white matter and were moderately reduced in the cortex. And these changes were observed in 2 second trimester subjects, but in 5 out of 8 third trimester subjects. We observed also changes in the subplate. The changes consisted in an excessive apoptosis or neuronal death confirmed by the immunohistochemistry by synaptophysin. Synaptophysin is a neuronal marker. In a normal, in control, third trimester subject, synaptophysin shows a diffuse labeling of the cortex, you can see here, with a particularly darker subplate, here indicated with the white arrows. However, in the 5 out of 8 third trimester subject, the labeling of the cortex was weaker, and particularly the subplate was thinner and paler compared here in the CHD subject and here in the control, confirming the neuronal death. The synaptophysin was also studied in second trimester subject, but none of the second trimester subject displayed excessive apoptosis, and synaptophysin labeling was strictly identical in second trimester subject with cardiopathy and second trimester control subject. Germinal zone injury was characterized by a reduced volume of the germinal zone compared here with control, and with the presence of pale areas of various size. And in these pale areas, we observed a significant cellular loss associated with diffuse astrocytic gliosis. We never observed such lesion in control, obviously. And we observed also Alzheimer type 2 astrocytes, which are typically GFAP negative, in globus pallidus, thalamus, and periventricular white matter. The Alzheimer type 2 astrocytes are a marker of hypoxia. Subject in our statistical analysis, subjects were classified according to their gestational age because we were interested in observing differences in the timing of lesion occurrence between second trimester and third trimester subject. We were interested in observing this difference because the third trimester is a particularly important neurodevelopmental period during the fetal life. Statistical analysis showed a significant difference in brain weight between second and third trimester subject, with a lower mean rank of brain weight in third trimester subject compared with second trimester subject. Statistical analysis detect also significant correlation between the gestational term and the presence of lesion in the white matter, subplate, germinal zone, and basal ganglia. In all this structure, the lesions were significantly more frequent or exclusively present in third trimester cases. No significant differences were observed for the mean rank between the different cardiopathy categories, and no significant correlation was observed between the frequency of brain lesion and the fetal sex. Our second objective in this study was to understand the mechanism leading to abnormal brain development. The changes observed in the hemispheric white matter in our third trimester subject were similar to diffuse white matter lesions described in encephalopathy of prematurity as delineated in numerous detailed neuropathological studies. Indeed, brain injury occurs in the same developmental critical period, which is the third trimester. The third trimester is a critical period during which connections are established and the first networks are formed in the brain, and particularly in the cortex, making the third trimester a period of particular vulnerability for the developing brain. In the injured white matter in our subject, the oligodendroglials, as previously reported, oligodendroglial cell density was identical to control, but numerous oligodendrocytes showed pycnotic or apoptotic nuclei, as you can see here, suggesting a particular vulnerability of this cell type. In the third trimester, pre-myelinating oligodendrocytes are the most prominent cells of the oligodendroglial lineage in white matter because mature oligodendrocytes differentiate later. Studies in preterm infant and animal models have shown that pre-oligodendrocytes are particularly vulnerable to hypoxia ischemia, making them one of the main targets of brain injury. In our injured white matter, we have observed activated microglia. Activated microglia release cytokine and glutamate, which are responsible for excitotoxicity. And this excitotoxicity is responsible probably also for pre-oligodendrocyte and white matter injury. Pre-oligodendrocyte injury is followed by a proliferation of progenitor of oligodendrocyte that restore the injured pre-oligodendrocyte, the dead pre-oligodendrocyte. But the new pre-oligodendrocytes show a failure of maturation and a lack of proper myelination capacity, leading to subsequently hypomyelination. Brain injury affects also components of white matter. We have observed significant axonal lesion in the crossroad areas, which are the area where motor, sensory, and associative pathway cross in 7 out of 8 third trimester subject and in only 1 second trimester subject. Periventricular crossroads are known for their particular vulnerability to hypoxic ischemic injury. And experimental studies performed in animal models have demonstrated that developing axons are particularly vulnerable to hypoxic ischemic injury. We observed also Alzheimer astrocyte type 2 in addition to stellate astrocyte, GFAP positive, in periventricular white matter and in thalamus and globus pallidus. Alzheimer type 2 astrocytes have been also reported in neonates with hypoxic ischemic injury in periventricular white matter and basal ganglia, and the presence of this Alzheimer type 2 astrocyte is considered as a marker of hypoxia. We observed also injury to the subplate. Subplate is a transient lighter in the transient layer, which is present throughout the mid-fetal and the late fetal period. It plays a crucial role in the development of early cortical connection and in the maturation of the neocortex. In addition, remnants of subplate are present after birth and provides the growth of short corticocortical pathway, which are essential for the establishment of cortical connectivity in the early postnatal life. The location of subplate between the periventricular white matter and the cortex close to the outer part of white matter make it vulnerable to white matter injury. We have observed in our 3rd trimester cases an excessive apoptosis, but this excessive apoptosis was never observed in 2nd trimester subject. This is consistent with a vulnerability of the neuron of subplates in this critical period, the 3rd trimester. The consequence of subplate excessive neuronal loss is an impairment of the development of cortical connection, leading to delayed cortical maturation, which subsequently will be responsible for cognitive deficit. And this subplate excessive neuronal loss contributes also to the impaired brain growth observed on prenatal and postnatal imaging. We observed a reduced number of calretinin interneuron in the injured white matter and also in the cortex in 5 out of 8 3rd trimester cases and only 2 2nd trimester cases. In humans, interneurons are produced in the germinal zone here in the ganglionic eminence and also in the dorsal ventricular zone, and they migrate from the germinal zone to the cortex through the white matter in the 3rd trimester, this period of particular vulnerability. Several others have described a deficit in interneuron in the white matter in preterm babies with perinatal injury. And these authors hypothesized that the interneuron in babies with perinatal injury are damaged because they migrate in an injured white matter. In addition, in our subject, in 7 out of 8 third trimester subjects, we observed a significant cell loss in the ganglionic eminence. GABAergic interneurons and all interneurons are produced in the ganglionic eminence in the third trimester. So the cell loss in the ganglionic eminence contribute in addition to the reduced number of interneurons migrating in the white matter to the cortex. Our third objective in this study was to correlate the lesion with antenatal imaging findings. Our statistical analysis have confirmed that the impact of congenital cardiopathy occurs in the third trimester, confirming imaging data published in the literature. After preoligonadocyte death, the new precursors do not mature adequately, and they are not able to perform a normal myelination, leading to an impaired myelination as identified in imaging. An injury to growing axons, subplate neurons, and migration of interneurons participate to an abnormal development of neocortical connection, and also contribute to the impaired development of neocortex, as observed in MRI in several publications. In summary, in this study, we have confirmed that the impact of congenital cardiopathy occurs in the third trimester. The lesion observed in our third trimester subject have demonstrated that white matter and its components, oligodendrocyte, growing axons, subplate, migrating interneuron, are the main target of injury because all of them are particularly vulnerable to hypoxia ischemia. The lesions observed in our subject with congenital cardiopathy are similar to those affecting preterm infants because they occur in the same developmental critical period, and probably they might share some common pathogenic mechanism. Indeed, neuropathological and imaging studies in normal subjects have demonstrated that a 4-fold increase in neocortex volume occurs in the third trimester, and particularly in the second part of the third trimester, due to a tremendous development of neocortical connection and late migration of GABAergic interneurons. This makes this period particularly vulnerable to injury, and particularly to hypoxic ischemic injury, because they occur in this particularly vulnerable period to injury is particularly hypoxic, due particularly to hypoxic ischemic injury in both preterm babies and in third trimester fetuses with congenital cardiopathy. Finally, we would like to emphasize the unavoidable limitation of our study. First, the unpredictable time span between fetal death and postmortem examination leads to a variable preservation of brains, and in some cases it's very difficult to interpret the lesion when the brain is, for example, macerated. It's difficult. Second, the difficulty in finding true controls cases, as finally, true control does not exist in theory, because fetuses or neonates with a normal brain, when studied after medical termination or perinatal death, have always experienced some degree of damage in utero or at birth. And finally, we have a statistical limitation, a clear statistical limitation, because we have only 8 third trimester subjects included in our samples, which is a small group, and the results are difficult to generalize to a larger population. Further larger and multi-center studies are required to support this finding and confirm the pathogenic mechanism underlying the relationship between congenital heart defects and brain development. Thank you for your kind attention.